Hello students, today we are going to get idea about very interesting point, that is a somatic hybridization. But in somatic hybridization, or before getting idea about the somatic hybridization, it would be a definitely necessary to understand about the isolation of the protoplast. Isolation of the protoplast, which is the most important first step of the getting idea, or just to go for the somatic hybridization or production of a somatic hybrid, isn't it? So for that one, we have to get idea about first of all the isolation of the protoplast, how to we have to produce, what is protoplast, these things, and then we will go for the somatic hybridization or production of a somatic hybrid using a vegetative cells or somatic cells. And this part is from your unit second, that is a plant tissue culture. Friends, as already I have said to you, what is the protoplast? Keep in mind, in case of the plant cell, cell wall is there. That cell wall is followed by plasma membrane. Inside the plasma membrane, we can find the cytoplasm content, and then nuclear membrane, then nucleus or nucleoplasm is there. So this is the general parts of the plant cell. Out of that one, suppose the only cell wall is dissolved, remaining part, that is nucleus, nuclear membrane, cytoplasm, and cytoplasm is plastered by the plasma membrane. Up to plasma membrane, the things are there, that is nothing but the protoplast. Keep in my mind, keep in your mind, my friends, because that is most important. Protoplast, plast means cover, cover of a plasma membrane around the cytoplasm, and inside the cytoplasm definitely there is a nucleus is there. So that total part without cell wall, or we have to without cell wall, or we have to say that naked cell, isn't it? Naked cell, that is cell is without cell wall, that is nothing but the protoplast. So there are the two methods of isolation of a protoplast: mechanical method and enzymatic method. These names itself of isolation methods tells us the which things are going on, or which type of procedures are taking place just to isolate the protoplast. In case of mechanical method, definitely skilled workers are required. We have to use the microscope, we have to use the sharp blade, sharp knife, just to cut or remove the cell wall, isn't it? After removing cell wall, the part of the cell, that is the protoplast, is there. Naked cell wall, mechanical method is there. Enzymatic methods means here we have to take a enzymes. Which are those enzymes we have to take place, how to isolate, so that we have to learn in this unit, in this lecture. Mechanical method, in this method plant tissue cell plasmolysis takes place, isn't it? Removal of a cell wall with knife, only cell wall, release of a protoplasm, and collection of a separated protoplasm of that. So protoplast we have to get. Now disadvantages of mechanical, as already I have said to you, here we have to use the skilled workers, microscopes, some instruments, and with the help of that one we have to isolate the protoplast from the tissue or from the cell. So here cell wall with the knife we have to remove. Skilled workers are required. Again, keen observations of them should be with that worker. So these things are there. So there are a lot of, we have to say that disadvantage of mechanical method: low yield of protoplast. Keep in mind, the worker who is a skilled worker, having his own, we have to say that, capacity, having his own limitations, and he would be able to give you a protoplast of a specific tissue, or we have to say that number of protoplasts he will be able to give you within a hour. Specific, we have to say that number of protoplasts he could be able to isolate. Means low yield of protoplast is there. Again, this is a tedious method, because you see that worker will have to work. If some worker is having a problem, again there is some problem, difficulties to the worker may be there. So that thing, so tedious method, low protoplast viability, that is most important, because at the time of isolation of the protoplast, many more injuries can take place to the protoplast, because that is mechanical method, that is depend upon the skill, which must how much we have to such skilled fellow is that who is dealing with the isolation of a protoplast is there. So definitely disadvantage of mechanical methods are low yield of protoplast, tedious method कंटाळवाणी आहे, लो प्रोटोप्लास्ट व्हायबिलिटी, कमी आपल्याला प्रोटोप्लास्ट हे वायबल मिळणार आहेत, बिकॉज फर्दर दॅट प्रोटोप्लास्ट वी हॅव टू यूज फॉर द fusion of them, and from that one we have to produce a hybrid, isn't it? So viability is most important of a protoplast that can be loses due to the we have to say that the misconduct or misbehavior, or we have to say that there is no sharpness in case of isolating protoplast. Now this is the method, my friends. I will show to you which is a mechanical method. Here epidermis is there, we have taken the cells of the epidermis, then that is plasmolized, isn't it? Means plasmolized cell is there with us. Now dissection of the cells, isn't it? So plasmolized cells, in the sense the cells is separated from this group, then dissection of cell has been taken, single cell is taken by the expert, and the cell wall, we have to say that this cell wall is removed, and this part is a definitely there is a protoplast, only protoplast is there, the outer covering that is of a plasma membrane, inside that one cytoplasm and nucleus is there. So this is the mechanical method, tedious, which is having a lot of disadvantages. Now just to overcome the difficulties, we know that in case of the research, in case of the each and every method, the first method is there in our hand, we are using that method, but sometimes just to overcome the difficulties of that method, just to make that method again superior method, or to develop another method, always scientists have been trying for that one, and this is again part, the enzymatic method is there. In this enzymatic method, definitely name gives idea to us. Here the enzymes are used to isolate the protoplast. Enzymes are used to isolate the protoplast. Here these steps are there of this enzymatic method. Sterilization of a leaf, in the sense we have taken the live leaf from the plant. Now the epidermis is removed of that one, then that is plasmolized, and the cells are treated with the enzymes, that is the pectinase, and another means mixture of enzyme is there, that is a pectinase plus cellulase. Definitely pectinase is responsible for the dissolution of a pectin, and cellulase here acts as a dissolver of a cellulase, isn't it? The cellulose pectinase that is dissolved, in the sense cell wall, contents of the cell wall are there, pectin and cellulase, these are the two major content of the cell wall. If that things are dissolved, definitely cell wall can get dissolved from the cell, and the released, that is a protoplasm, means we will be able to say that protoplast is in our hand. So enzymatic method having a lot of, we have to say that, importance or significance of this method as compared to the mechanical. So nowadays in the research, mechanical method is not used, only enzymatic method is used. Why? So we have to see the advantages of that one, isn't it? Which are the advantages of enzymatic method? Here, my friends, you have to keep in mind, it is applicable to variety of tissues and organs, including petioles, fruits, roots, coleoptile. So little bit there is a no bar, the tissue of which part of the plant is to be taken for the isolation of the protoplast. So there is a variety, isn't it? There is a variety of things. We can isolate the protoplast from any part of the plants that is there. So that is most important, we have to say that advantage. First advantage of a enzymatic method of isolation of a protoplast is there. Now mesophil tissue, most suitable. So in case of enzymatic method, suppose in our hand we are having a mesophil cell of a specific plant and mesophil cells of another plant, in between which we want to or with the help of that two, we want to produce a hybrid variety. It is possible. So mesophil cells of the two different but desired character having plants we can take. That is very convenient method. High yield of protoplast is possible. if we'll take the mesophyll cells of that one. Again, easy method. More protoplast viability is possible, which is not possible in case of a mechanical method. So this is a little bit different between the mechanical and enzymatic method, and because of that one, most of time for the isolation of a, not most of time, I will tell that 100% now cell wall is dissolved by using the mixture of a two enzymes, that is cellulose and pectinate, and because of that one we are going to get the higher yield. Easy method is there, more we will be able to get, and in speedy process is that one, within a short of time we can get a more than enough. We have to say that protoplast can be isolated with the help of this enzymatic method. There is no need of a skilled workers, means skilled worker is required, but the worker who is expert in removing of a cell wall is not required here, isn't it? Only the fellow who is having a good observations or keen observations, or who will be supervise the enzymatic method process very well, that type of skill worker is required here. Now my friends, as I have already said to you, you have to look at this diagram. In this diagram we will be able to see that protoplast of A strain, protoplast of B strain is there, isn't it? And here the fusion treatment is given to that one, then adhesion takes place, fusion took place, and fused protoplast we will be able to find out with heterokaryons, in the sense two nucleus are there, one from A strain, another from B strain. Now this A and B fuses with each other, and finally we forms the cell which is a hybrid one, isn't it? So nucleus get fused, protoplasm of both the two different strains get fused, and finally we are getting the cell that is a hybrid cell. So this is the diagrammatic representation of a protoplast fusion. As we have seen, the two methods are used here, mechanical method, enzymatic. Out of that one we have given the preference to the enzymatic method. We have isolated the protoplast from two different strains having a plant. Now that are we have fused with each other with the help of a fusogen, means the some chemicals are there which are inducing fusion in between two strains protoplast. That's why that are known as a fusogen. When fusion will take place, the single cell will form, and that cell is a mixture of, we have to say that, two nucleuses and two protoplasm, isn't it? So fused protoplast, heterokaryon condition, that two nucleus get fused with each other. So hybrid we will be able to produce with the help of a fusion of a protoplasm. Vegetative cells we have taken here, isn't it? That you have to don't forget. Now the main, again we have to say that point from this syllabus point of view, that is a somatic means we have to say that vegetative cells, there is a hybridization process takes place in between the two vegetative cells here, isn't it? The steps which are involved into that one, isolation of the protoplast, as we have seen in detail about the what is protoplast, then which methods are used for the isolation of protoplast, then how it is to be fused, that we have studied. So in that one, first method is isolation of the protoplast. Suppose we have taken the enzyme help of an enzyme and isolated the protoplast from the two different cell. Now then fusion of protoplast, the two different type of strains protoplast we have isolated, and that then we have to fuse it, but with desired species, isn't it? Because the hybrid whatever we want to develop, that could be shows presence of a desired characteristics. In the sense, I will tell to you, suppose we want to produce a hybrid, somatic hybrid of a mango plant, then one mango plant having a sweetness, another mango plant is a larger in size, isn't it? The sweetness having mango plant is a smaller in size, we want to larger in size, but here in another mango another strain is with us. So like that one, two desired characters are there, sweetness and larger size. We want to combine that one, and then we have to produce the hybrid with the help of a vegetative cells. Protoplast we have selected from the vegetative cells with a having desired species of that. Now identification and selection of the hybrid, again most important. That's why skilled workers, I have tell to you, here also skilled workers are required, but the job or role of that skilled worker is a different here. Now after selection or identification of a proper hybrid cells, culture that hybrid cells on a culture medium and development of this hybrid into plant. Here we have to write develop into a plant. So that is possible, isn't it? Culture of hybrid cells and develop into a plant. So these are the steps involved in the somatic hybridization. Here my friends, I will give you idea. Here you will be able to see the procedure, what things are going to happen here. Now here we have taken into hand our, first of all, the young plant, isn't it, having a desired characteristics. Now that is to be, first of all, we have taken here plant material, that is a leaf, but that material we have to sterilize first of all, isn't it, because we have to grow this plant material in a culture medium in septic condition. So in the beginning, from very beginning, we have to be a little bit careful just to kill the microbes. Here the leaf is to be sterilized. After sterilization of the leaf, the epidermis is to be peeled out. Remove the epidermis. After removing epidermis, we will find a such a type of a peeled leaf segment, that is to be plasma. Then it is to be plasmolized. So we will get able to such a type of two cells. Now cells in enzyme mixture. Put those cells into the cellulose and pectinase enzyme containing mixture. Put these plasmolized cells into the cellulose and pectinase containing mixture. Then after few or incubation or after few hours, you will be able to find out cell wall, digestion and release of protoplast takes place. So here we will be able to say that upper layer protoplasts are there, and at the bottom we will be find cell debris are there. Now this protoplast is to be isolated. Plating of protoplast, this protoplast we have plate, cell wall generation takes place, means then clumps of the cells, callus culture, differentiation of a callus culture and plantless is derived from a protoplast. So what happens actually here in this method, we will be able to see that we have taken a plant part, that is sterilized. After sterilization, we have plasmolized. After plasmolized, we have put that plasmolized leaf part of a plant into a into a container containing mixture of a cellulose and pectinase enzyme. Then what happens actually after 2-3 hours or 4 hours, that is depends upon the which type of plant material and which type of plant is selected for the protoplast isolation. Now we will be able to get the protoplast, and then isolated protoplast we have to plate on the put on the culture medium, and then again what happens actually on that culture medium, cell wall get formed around that one. After cell wall forms, the multiplication takes place, clump of many of cells can get developed, final callus, and from the callus differentiation is possible, and from the differentiation of a callus tissue, plantlets derived into the that culture medium containing tube, isn't it, and culture tube. So like that one from this protoplast. So major steps involved in a protoplast isolation, culture and regeneration of plants. Friends, keep in mind in this diagram only we have isolated the protoplast of a single cell, and that we have cultured on a culture medium, and finally we have produced the plantlet only of same plant. Here hybridization has been not taken place because for that one different type of protoplast are should be there. Now this diagram will give you idea about somatic hybridization. Here we will be able to say that species A, which is green, species B, albino or non-green. So here from the species A we have isolated protoplast, species B we have isolated protoplast. Now this isolated protoplast from the different two variety of a plant is fused here, fusion of those green and albino. Now fusion will takes place. Now cultured on the selection medium. Now we have to find out the exact very we have to say that interesting or proper we have to say that hybrid cells fusion will takes place. Once the fusion will take culture on the culture medium, or we have to say that selection medium, then we will be able to find out the one culture we will get from that one, that is a small green cell colonies only, green cell colonies only, and white cell colonies only, isn't it? So that you have to keep in mind, small green cell colonies, green cell colonies. Now no further growth will be takes place here. Here green callus will be added. Somatic hybrid we have developed. So fusion has been done in between the two protoplast of two different, and then fused. Then we found the hybrid of a green cell colonies, that from green cell colonies which we have selected after selection, again we have grown that one on a culture medium. We have got green callus, that is undifferentiated mass of the tissue, and then finally we have grown converted into the somatic hybrid. So this is the diagrammatic representation of a selection of a hybrid coupled with the selection of coupled with the growth on selection medium. So this is the method of selection of all these things and somatic hybridization. Now protoplast fusion, here double we have to say that the word is that protoplast fusion. How to achieve? So there are again methods of protofusion are spontaneous and induced. Which are those? Those are spontaneous and induced. Spontaneous is one, induced is one. In the sense, spontaneous means naturally protoplast fusion takes place, that is spontaneously. You know that most of times the question is to be asked in the classes and students spontaneously answer for that one. And sometimes what happens is some students are there, we have to induce them, please stand up, then you will have to tell compulsion, so induction is there. Here protoplast fusion spontaneously takes place, willingly in between these two protoplast fusion is possible. There is no need of external forces or use of some chemicals. In induced diffusion, induced fusion we have to say that, not diffusion. Spontaneous or induced fusion is there. Induced fusion. So spontaneous fusion method in this cell fusion is a natural process as observed in case of eggs fertilization. Generally in the we know that actually male gamete and female gamete, isn't it, fuses with each other, isn't it? So that is a spontaneous process in the nature occurs, means egg is fertilized by the male gamete. So that fertilization process in the nature takes place, that is a spontaneous one. During the course of enzymatic degradation of the cell wall adjoining protoplast may fuses to form a homokaryocytes. They form the homokaryocytes. The cell contains high number of nuclei in that one. Cell contains high number of nuclei, that is 2 to 40. We can find the fusion of spontaneous fusion, we can find, and most of times in single cell we will be able to find out more than 2 to 40. That is the range how much nuclei can get or how much cells can get fused with each other. So in protoplast fusion, the spontaneous fusion we have seen, the protoplast fusion can be achieved by spontaneous fusion or induced fusion. This same slide is there. Now in case of induced, as already I have said, we have to make them, we have to force them to get fused, but for that one we have to use some chemicals, and whenever we are using some chemicals to fuse the protoplast, and that chemical, that agents are known as a then fusogen, isn't it? We have to use some force, we have to use some chemicals, and that chemicals are nothing but the again we have to known as a fusogen, because they are playing role of a fusing protoplast. That's why they are known as a fusogen. They are inducing to fuse the protoplast, so that they are collectively known as a fusogen. Which are those chemicals? KNO3, potassium nitrate, glycol, polyvinyl glycol, isoenzymes, electrofusion, dextran and dextran sulfate, fatty acids and esters of that. So these things you have to keep in mind. What is actually we have to make them to get fusogen. Now what are the advantages, my friends? We know that in the nature or most of times there is a fertilization process is there. Young is egg fuses with the male gamete and zygote develops, and that fertilization process further goes into develop into a new plantlet is there. But in case of this, so there is a reproductive cells, but here we have to keep in mind vegetative cells we have used to develop the new variety. Vegetative cells of A strain, vegetative cells of B, we have taken. Now from the A strain we have taken the A protoplast, B protoplast from B, and fused that A and B protoplast and heterokaryocytes that is developed, and that is further developed into a hybrid, further developed into a plantlet, that is a somatic hybrid plantlet. So what are the advantages of that one? It is production of a novel interspecific generic hybrid. Pomato, very interesting, my friends. Actually pomato in the sense single plant is there. Below the ground, that plant, that variety produces or develops potato, and above the soil part we will be able to find out the fruits are there. So single plant produces potato and tomato, single plant. So this type of a novel interspecific intergeneric hybrid. So this hybrid has been made in between the protoplast isolated from the potato and protoplast isolated from tomato. Fused that two protoplast with the help of a fusogen. Then further development into a pomato plantlet. Actually this is right now not available in the nature, because most of time you have to keep in mind, I used to say that nature has a final right to give a chance to organism either to be on the earth or that should not be on the earth. So final decision is of a nature. So this variety, such a type of plants are developed by the scientists, succeeds in the development, but this variety only could be able to survive in the laboratory condition, in the controlled condition. But outside the nature, outside the laboratory, this plant never grown, because that is not accepted by the nature itself. So nature also has a right to accept and reject the organisms, isn't it? So these things are there. Pomato variety, production of a novel that is possible with the help of somatic hybridization. Then production of a fertile diploid and polyploids from the sexually sterile haploids, and diploid and polyploid from the sexually sterile haploid, and any aneuploid. So you have to keep in mind, my friends, most of the time there is some plants are not able to produce the male gametes and female gametes. When no production means definitely we have to say that they are sexually sterile. But in case of such a type of sexually sterile plants, it is possible, isn't it? It is possible to develop a new variety with the help of a somatic hybridization. The transfer of gene of a disease resistance, abiotic stress tolerant, herbicide resistant, production of heterozygous lines in the single species which cannot be propagated by a vegetative means. We know that most of the plants are reproduces with the help of a seeds, means sexual reproduction is required, isn't it? But in such a plant, naturally vegetative reproduction is not there. So they are not able to produce a new plantlet with the help of their parts, vegetative parts. In such a condition, the somatic hybridization have a capacity, this technique can be used just to develop a vegetative, means by taking a somatic cells, vegetative cells of a sexually reproducing plant, we can take a vegetative cells and develop a new plant which are having various type of a genes. Suppose A variety having a disease resistance variety and B is a disease susceptible. If we'll fuse the cells of these two, protoplast of these two, then we will be able to develop a C variety, hybrid variety of a disease resistance. Same condition in case of abiotic stresses. We know that many more abiotic means the stresses which are caused because of a abiotic conditions, abiotic factors means temperature is there, isn't it? Temperature, then some gases, abiotic stresses are there, isn't it? Cold stress. So this type of abiotic stresses which are not created by the, which are created by the non-living things, abiotic stresses are there. Herbicide resistance variety we can develop, isn't it? So you have to keep in mind the somatic hybridization has a lot of importance. Now studies on the fate of plasma genes, we can study the genetic study is possible with the help of a somatic hybridization. Then production of a unique hybrid of a nucleus and cytoplasm. This is also one of the most important advantage of a somatic hybridization. But some limitations are there. No doubt that we have to say that small, we have to say that difficulties we can find sometimes with the help of this somatic hybridization. That is a poor regeneration of hybrid plants. The developed hybrid cells or plants may not be regenerate, isn't it? The non-viability of fused products. Although we have fused the two different type of a desired characters having protoplast, that may be the hybrid whatever we have got, or heterokaryon containing cell is there, that may be non-viable. Not successful this method in case of all plants. Some plants, in case of some plants, somatic hybridization fails to produce the somatic hybrid. Production of a unfavorable production. Unfavorable is sometimes whatever we will get the plantlets in our hand or hybrid. After taking a proper care, each and everything we have taken at every step we have looked with we have to say that 100% concentration we have given to every steps of production of a somatic hybrid. Then also finally the plant whatever we have got, whatever hybrid we have got in our hand, maybe the hybrid of a unfavorable, we have to say that, in the sense all the desired characters may not be, we have to say that, get expressed into that one. So that is also there. This is also one of the limitation. Lack of efficient method for the selection of hybrids. So there are very few methods are there which can be used for selection of a proper hybrids. No confirmation of expression of a particular trait in a somatic hybrid. As already I have said to that, we know that when we want to introduce a herbicide character into the hybrid, somatic hybrid, at that time that may there is no confirmation, there is no guarantee that may can express into the somatic hybrid whatever we have developed, isn't it? So some limitations are there of those things. Now some applications I will tell to you of this somatic hybrid. Very useful methodology, very useful hybrid producing methodology is this one. That is, we can produce by manipulating the genetic conditions of this one. Disease resistant variety we can produce the environmental tolerance, quality characteristics we can be able to produce. Cytoplasmic male sterility is there, so that we can remove with the help of this methodology. Again, to study the cytoplasmic genes, functions of the genes we'll be able to study with the help while going through this hybrid production method. So the information is successfully useful in plant breeding programs. The whatever information we will get of that protoplast, of that naked cell, that is useful in case of other type of breeding programs. Now it helps in the combination of a mitochondria and chloroplast. So we know that actually the genes are present in mitochondria also. Some genes are present in chloroplast also. So when we are going to fuse a protoplast of a two different protoplast from the two different plant, definitely at that time combination of mitochondria of A and B, combination of a chloroplastic gene from A and B variety, and that could be result of a unique nuclear cytoplasmic genetic combination. That is also possible with the help of a this somatic hybridization technique, technology or method. It can be done in a plant that are still in a juvenile phase. We can There is no need to take only the plant parts or isolate the protoplast from the plant parts of a mature plant. In juvenile phase also it is possible to do this somatic hybridization. Protoplast transformation is followed by the somatic hybridization. We yield innovative plants. Innovative plants will be able to develop in our life. We know that actually the most of times we are adding a urea into our field. A farmer used to add urea into the field, means more and more nitrogen is provided for the proper growth of the plant. But we know that there are many monocots, for example jowar, bajra, wheat, rice. These plants are there, they are unable to, we have to say that, synthesize the nitrogen on their own, isn't it? On their own, although 70% nitrogen is available around them. So in such a condition, suppose we could make the sugar variety, the jowar variety, bajra variety, to fix the atmospheric nitrogen by their own, isn't it? By adding nitrogen fixing gene into those plants. So that is also possible, my friends, with the help of this somatic hybridization, isn't it? So very interesting and nice this somatic hybrid methodology is there. Today we have learned, my friends. First we have got idea about what is the protoplast. Again I will tell to you, concentrate on this question. What is protoplast? Isn't it? Which type of methods are used for isolation of a protoplast? Which is a superior method of that isolation? Isn't it? Then fusion is there. What is fusogen? This question also you will have to answer. What is fusogen? What are the examples of the fusogen? Isn't it? What are the steps involved in the somatic hybridization? What are the steps involved in the somatic hybridization? What are the applications of a somatic hybridization? Then what are the limitations? What are the advantages? All the points you will have to get it covered. All the points you will have to note down. Read each and every word very carefully. Then you will come to know about the what is protoplast, the methods used for the isolation of protoplast, the what is fusogen, what are the types of fusing of a protoplast, just like one spontaneous method of fusion of a protoplast and another induced one, isn't it? And finally all the things you have to learn. With this thank you